Welcome back. Welcome back, everybody. Thank you very much for the invitation to come and give the second half of a talk that was obviously too long for just last time. So as Frederick said last time, I talked about loaded and unloaded swallows. So if you're a Monty Python fan, that should amuse you. But in the context of the response of the Aplysia motor system to engaging different external loads. And today I'm going to talk about what to me seems like a parallel set of problems in understanding how the mammalian respiratory system adapts to different metabolic demands. So I'll skip some of what I've done is I've updated the slides I had last time. Oops, I went too far. Here we go. Yeah. So we talked a little bit last time. We started with a discussion about reasons why one would want to study brain circuits as they are connected to interactions with the body and through the body to the environment, both the pros and the cons. So I won't recapitulate that whole discussion now, but for those who weren't here last time, there were plenty of reasons why one might want to study the brain or parts of the brain in isolation, experimental control, lower dimensionality, simpler just to think about, mathematically simpler for modeling. And on the other hand, the real world is the brain co-evolved with the body, strongly coupled to the body. And so what I hope to convince you of today is that in the mammalian breathing system, which is a system none of us could do without for very long, although the mechanisms by which the respiratory oscillation is produced in the isolated system, in vitro, so to speak, are well understood and have been understood for pretty well for a long time, the mechanisms in the intact system, in the way it actually works in the body, aren't necessarily the same as the way it works in the isolated system. And in fact, I'll try to convince you that they're different. Okay, so let me jump ahead now so last time we voted which example system to look at. And last time we looked at the sea slug. So now we're going to look at mammalian respiration. On the top right is an experimental figure from my collaborator, Chris Wilson at Loma Linda University. And the sort of data he's collecting is he can record the respiratory effort or the changes in pressure produced by the lungs and the breathing system in, say, a rat. He's doing whole body plethysmography, where you take an animal and you put it inside a chamber where, as it breathes, the chamber is a little bit flexible, and you can actually measure the pressure differences that it's producing as it's breathing. And one of the interesting things that once you think about it, everyone has experienced this, but usually you don't think about breathing. If you're just sitting quietly listening to this talk, you may get sleepy or something, but if you're not getting up and walking around, your breathing will be pretty regular, but every once in a while you'll make an augmented breath, an extra deep inhalation, a sigh, or just a sort of luxurious, extra large inhalation. And we see that also in the mammalian breathing system here, this very large spike. So this is regular breathing, what we would call eupnea. And then occasionally it's interrupted by this very large inward breath. I don't have a lot to say about that in this talk, but we have a grant under review where one of the things that we propose to figure out is what's going on with those. So I just thought I would put that up there at the beginning. But now I'm going to step back a couple of decades to when the breathing, the area of the brain that is responsible for producing the most basic part of the breathing rhythm, which is an inspiration, an inward drawing of breath in a periodic fashion, that was isolated in a landmark series of experiments done by Jeff Smith, who's at NIH. Basically, what they were able to do is, so this breathing rhythm is being generated in the brainstem, and you can dissect out the brainstem. Here's the brainstem dissected out, and you can take out this little piece of it down here, and you can just cut slices of it away. You keep cutting it away until it stops breathing. By which I mean, you can put an electrode in here and record, and you see what they're called fictive breathing patterns. So you see the particularly easy nerve to record from is the 12th nerve, which is the one that the hypoglossal nerve. When you breathe, several things have to happen at once. In addition to your diaphragm pulling down, the back of your throat pulls up, then your tongue gets pulled out of the way. The nerve that's driving that is particularly accessible, and so you can measure the timing of respiration by looking at the output of the nerve that would be going to pull the tongue back. And so you can trace things back to this one little nucleus called the pre-Butzinger complex. It's just a few hundred cells on each side, 600 or so on each side of the brainstem, and it produces this reliable, regular breathing rhythm or fictive breathing all by itself. Here's an image of the slice through the brainstem and the recording of the activity on the nerve that they're able to access. Okay, so this was a big deal in 1991 when it first came out. Within a decade, John Rinzel and Rob Butera had gotten together with Jeff Smith and put together a conductance-based model based on more detailed electrophysiological experiments, whereby combining persistent sodium currents and a few other standard currents, they were able to reproduce the laboratory observations about these regular breathing rhythms. So I'm going to take a moment now to introduce you to this model for those who aren't familiar with it. And you know, there is a chat function here. If you want to pop a question into the chat, I will try to keep an eye on that. Or you can unmute yourself and tell me to go faster or slower or whatever at any point. That would be fine. But okay, so it's a standard conductance-based model. This is the so-called Butera-Rinzel-Smith model. There's a standard sort of inwardly rectifying potassium current like you would see in Hodgkin-Huxley, a fast sodium current like you would see in Hodgkin and Huxley, a leak current. And then there's two additional currents. This persistent sodium is a slow excitatory current. And its gating variable, what makes it persistent is that the H here is a very slow gate. It inactivates very, very slowly. The time constant for, say, the N in the potassium channel is 10 milliseconds. The time constant for the H current is 10 seconds, so 1,000 times slower. Okay, the gates follow standard first order activation and inactivation kinetics. They've got a sort of sigmoidal onset curve. The other new current is the G tonic. This is just an excitatory conductance that tends to drive the cell to fire, and that's going to play an important role later on. Okay, so right, so these are two of the important parameters, the G leak and the persistent sodium current. So in the original model, what Butera, Rinzel, and Smith were able to show was that this combination of currents could reproduce several different behaviors that were all observed under different conditions. So for example, so here we're looking at what happens as you vary the reversal potential for the leak, as well as the size of the conductance associated with the persistent sodium current. So if I make the, if I reduce the reversal potential for the leak, that's just like adding inhibiting or a hyperpolarizing constant current. So if I tend to suppress the cell, if I move this way, or if I reduce the strength of the persistent sodium current, then I end up in a silent phase in which the cell just sits at its resting potential and it's nothing happens. If I make the cell more excitable by either by increasing the leak reversal potential or increasing the persistent sodium conductance, then I can move into this bursting regime where the cell will fire a barrage of spikes, and then eventually the persistent sodium current inactivates, but it takes a while. So this is this time bar here is a couple of seconds. So the burst lasts a second or two, and then is followed by a gap of several seconds. And so this is the fictive breathing. This is consistent with what they observed in the laboratory. Now I can regulate the frequency of breathing if I change these parameters. So for instance, if I inject a little bit more current into the cell or disinhibit it a little bit, then the bursting is going faster. So here you see there's a breath maybe every 2 or 3 seconds instead of every 10 seconds. So there's a pretty big range of breathing frequencies. And of course, physiologically, you know, if you as you listen to this talk and I drone on and you get drowsier and drowsier and more relaxed, your breathing may slow down and you may gradually, you know, move somewhere along this diagram. If you decide to get up and go for a walk afterwards, then as you take a brisk walk, gradually your breathing rate will increase. So that's some aspect of that is captured here. Now, if you push things too far and you drive the cell too hard, or you make the persistent sodium conductance too low, then you end up getting stuck in this beating region. In the beating region, the cell fires a barrage of spikes, but it never inactivates. It just keeps on going. So this is just sort of like getting really excited and firing a lot of spikes. But in the context of breathing, this is not a good thing, because what this system, the way this is interacting with the motor system ultimately is there's a motor output. And when you take a breath, you want to have all of the motor neurons, you know, getting stimulated for long enough to inflate your lungs. And then you want them to stop so that you can relax your lungs. And you wouldn't want to be like just constantly breathing in and never breathing out. So the beating region would be pathological if it appeared in a, you know, in the intact system. And the bursting we would call eupnea. And there is a pathological form of rapid shallow breathing called tachypnea, which I'll come back to later. I'll spell it for you here. Tachypnea. And I'll put that with some question marks. It's, you know, you can interpret what's happening in a model like this in different ways, but one possible interpretation is tachypnea. All right. Are there questions? This is the beginning of the modeling framework. So this is a good time to ask questions if there are any questions.
Is there any, I have a question about hyperventilation. Is it something that like you're getting in a different regime and get stuck there?
Yeah, we're going to, so right now this is the isolated circuit. And what I will show you in a moment, well, a little bit later, we're going to embed this circuit in a closed loop respiratory control model so that we're actually driving the lungs and driving gas exchange. And there's going to be some sort of feedback loop where chemosensation you know, comes back in to drive the central pattern generator. And in that situation, what we discovered is that this model is capable of getting stuck in the rapidly beating regime, which would correspond, which we think might correspond to tachypnea. Okay, so yes, the answer is yes, and I'll get there in a moment. All right, so we wanted to look at the question of whether or not the mechanism that generates these oscillations in the isolated system was necessarily the same as the mechanism that generates the breathing oscillation in the intact system. As theorists, our way of doing that is to build a closed loop control model and see how it behaves. But let me point out a sort of paradox or a sort of central conundrum that is part of what motivated this work. So this sort of, all these systems that I showed here, or at least these ones that are oscillating, they form limit cycles, right? As a dynamical system, as a system of coupled ordinary differential equations, that's an example of a limit cycle. And just for those who aren't like doing dynamical systems all the time, let me remind you what a limit cycle is. So if I've got a system of differential equations, this is in some number of dimensions bigger than 2, or 2 or bigger. This is an autonomous system, meaning there is not an external, in these models, there's no external drive pumping the system, like some other brain area that's sending a signal. The isolated central pattern, the isolated pre-Bussinger complex is capable of producing this rhythm without any external stimulation. And so it's natural to model it as a limit cycle. So a limit cycle is a closed, isolated periodic orbit. Which can occur in a nonlinear dynamical system or nonlinear system of ordinary differential equations. So it's a solution of the equations. I'll call it little gamma of t. And it's periodic, so there's some number, big T, which is the period. Which, after which it repeats. So, all right. So the nice thing about limit cycle dynamics as models for central pattern generators, whether it's central pattern generator involved in locomotion or swimming in the lamprey, or last time pulling food in during swallowing, or the heart beating, or in this case, breathing, since they're autonomous and they're stable, you just keep doing the same thing over and over again very robustly, right? That's a good system to have if you're designing a breathing system. On the other hand, you want to be able to adjust the cycle, because if you do get up from your chair and go for a walk after this, you don't want to be stuck in exactly the same breathing pattern when you're trying to walk around as when you're sitting still, because your metabolic demand will be larger because you're exercising, and you'll need to have a variant of the underlying rhythm. So somehow we need to have the robustness that comes with an autonomous limit cycle type system combined with the flexibility to adjust to changing environmental conditions. Okay, so part of what motivates this is all this work is how do we understand that in real systems? So for the particular model that I'm going to tell you about, continue telling you about here, we take the Butera-Rinzel-Smith model that I sketched for you a moment ago. We build it into a closed loop control system where we try to make it as simple as possible while still having all the basic elements. So we're going to have lung mechanics. So the output of the Butera-Rinzel-Smith model is going to be driving a very simple muscle model that will open the lungs, pull air in. The outside air mixes with the air in the lungs, and then the oxygen from there gets pulled into the body, and then gets pulled out because of metabolism. And then to close the loop, we have some form of chemosensation. There are two main routes by which the brainstem responds to changes in blood gases: oxygen levels and CO2 levels. CO2 levels are a much more powerful and direct sort of mechanism. That's probably the most important mechanism. However, the experimentalist that I was working with was an expert in the oxygen side and the response to hypoxia. So in the model, we chose between the two. We wanted to keep the model as simple as possible to look at this fundamental question. And so we went with hypoxia chemosensation instead of hypercapnia. There's a series of beautiful papers, very good papers by a group involving Ilya Rybak and John Rubin and Slava Molkov and a bunch of other people, where they were looking at the hypercapnia or the CO2 pathway. So if you get interested in this area, I encourage you to look at both our papers, which are based on, our one paper so far, based on hypoxia or oxygen sensitivity, as well as theirs on hypercapnia. So we use oxygen sensitivity for our chemosensory component. And then I'll just tell you now what the main results are. So we can have an open loop model in which we just get rid of the chemosensation. We don't have the feedback loop closed. And then we see the intrinsic properties of the Butera-Rinzel-Smith central pattern generator model. And we can ask, how would the body behave if it were being driven by that without the sensory feedback? And we can look at the closed loop model where we also have the sensory feedback. And it turns out that for this relatively simple system that we have, you can show without any real doubt that the fundamental mechanism that generates the oscillation is different in the two cases. It's a different underlying kind of bifurcation, and the sensitivity, if you change different parameters, is different in the two cases. And I'll take you through some of the logic of that. You can read all about this in our paper that appeared in the Journal of Neurophysiology a couple of years ago now. We are actively looking at this model, Casey and Chris and I, now that everyone is thinking about COVID. One of the things that happens with the coronavirus, of course, it's a respiratory affliction, but there have been reports of people who are walking into the COVID clinic or the emergency room, and when they measure their blood oxygen, their hypoxia levels are so extreme they shouldn't even be conscious. Normal levels of oxygen in the body are like, or in the blood, are like 100 millimeters of mercury as the partial pressure of oxygen. For those not steeped in this stuff, the partial pressure of oxygen in the air that we all walk around in is normally 160 millimeters of mercury, and then it gets drawn into the lungs, and some of it gets taken out. And anyway, the air, the concentration in the blood is normally about 100 millimeters of mercury. So there are people with like 50 millimeters of mercury, walking around not realizing how sick they are until they suddenly collapse. And there are people who feel fine one day, and then they collapse the next day, and then the next, they're immediately on a ventilator. Some people coined this doesn't happen to every COVID patient, but some of them. Some people coined this condition the quote, happy hypoxics, although the community quickly condemned that use of language. Although it's colorful, it's not, these are not happy people. It's called silent hypoxemia or persistent hypoxia. And of course, if your oxygen levels are low, there's, for long periods of time, there's all kinds of brain damage that can contribute to. So it's potentially a serious problem, possibly related to the people with long haul COVID symptoms. Okay, so anyway, this is a 4-year-old paper, but we're looking at it afresh because of that. All right, so how do we close the loop for chemosensation? Well, we take we're going to have the blood oxygen as part of the model. Oops, I'd like PaO2, the alveolar concentration of partial pressure of oxygen is going to be part of the model. And what that's going to do is going to go through it's going to be fed through a sigmoid function. So here we have PaO2, and then we have our tonic conductance. So this tonic conductance here is going to be what closes the feedback loop. So when the oxygen level is low, the conductance is high, and it's going to support bursting or spiking. And then as the oxygen concentration goes up, the conductance drops gradually. And then if it drops down again, then it goes back up again. So there's a monotonically decreasing relationship between the strength of the drive and the O2 level in the blood. Okay, so here's a picture of our model, a diagram. We've got the central pattern generator with 3 variables: the cell voltage, the persistent sodium gate H, which is, remember, very slow, and then the potassium, which is relatively fast. The voltage from that is fed out to a simple motor neuron pool, which just, when you're spiking, it drives up the activation of the muscle, and then when you're not spiking it, the activation of the muscle drops, just as following a linear ODE. The muscle is driving the lung volume, so the lung is just an elastic, like a piston with an elastic spring. So it gets pulled to larger volumes. When you're doing that, fresh air comes in, it mixes with, we just assume it's well mixed, which of course is a terrible assumption, but we're trying to keep things simple. So you have the gases mixing in the lungs. The oxygen in the lungs transfers through the walls of the alveoli into the blood, and so we have the lung oxygen then some of it goes into the blood oxygen just by a diffusive process. And so during each breath, the blood oxygen goes up, then it drops down, then it goes up, then it drops down as you keep on bringing fresh air into the lungs like a bellows. G-tonic is doing the opposite. So as this goes down, this goes up, following that sigmoidal curve. And that G-tonic is then driving the tonic current that is an excitatory input to the central pattern generator. Okay. There are equations. We could, if anybody wants, we can go through all the equations. I'll just point out the form of the sensory feedback here that I sketched with that sigmoid is here. So this is, again, this is G-tonic as a function of the partial pressure of oxygen, and it follows this such a curve. The reason I point that out is if there's time, I can tell you a little bit about our preliminary results on modeling the effects of COVID on this, on the system as seen through this model, which the question is how could you be sustaining a low level of blood oxygen and kind of not realize it? Like, what parameters would you have to change within the model so that it mimicked what's observed in these silent hypoxemia patients? Anyway, that's something that we're working on right now. But before I get to that, I will I'll tell you about the way the model in its original form, the way it works. Okay, so we can run this model in a couple of different ways. The closed loop is what I just described. So G-tonic, that's the excitatory drive to the central pattern generator. That's varying as a function of the concentration of oxygen in the blood. The H gate, the persistent sodium gate, that's also dynamically varying just as part of the model. Now, the reason I'm interested in the H gate is because the slow time scale of the persistent sodium current gate is what sets the timing of the breathing rhythm in the isolated system. And I'll show you a plot in a moment, but if you take that time constant and you change it, you speed it up, then the breathing gets faster. And if you slow it down, the breathing gets slower, with all the other parameters being the same. So the timing of breathing, among other things, is related to the timing of the changes in the H variable. In the closed loop, you also have the dynamically varying G-tonic. And one of the questions we will ask is which of those two actually controls the timing of breathing in the intact system where they're both present. So to get at that question, we can play around with a couple different versions of the model. We could freeze G-tonic at the same parameter value that it would have had in the original open loop Butera-Rinzel-Smith model. That is to say, it has a nominal value, 0.3 nanosiemens. We just fixed that as a constant. Then we're looking at the original Butera-Rinzel-Smith model, and that's a way of doing open loop. We can also take a closed loop model where we allow the feedback to come back, but we freeze the value of the H gate. So we don't let the inactivation level of the persistent sodium channel change. And in that case, we can also get a breathing rhythm. So that's already suggesting that in the closed loop model, the mechanism that drives the timing of breathing is different. Because if you take the variable that normally controls the timing and you make it constant, as you see here, the H gate is just constant, you can still breathe. So how is that possible? Well, as the blood oxygen goes down, the tonic drive goes up, and now you've got the isolated system, the Butera-Rinzel-Smith system is no longer autonomous, and now it has a time varying input that can trigger it to fire. So you see that G-tonic is going up and down, and you get nice eupneic bursting. So there's different mechanisms here that all give similar kinds of bursting patterns. Okay, so to dig a little deeper into this, we asked, well, what actually controls the timing, or different aspects of the timing? So in this top plot, you can take tau H, which is the that's the time constant for the persistent sodium currents, and you can make it slower by rescaling it. So we introduced this scaling factor, and if you slow it down, then the interval IBI is the inner burst interval. The spacing between the bursts gets longer, and that's true in the open loop system. But in the closed loop system, where you're getting the sensory feedback, basically no change happens, and you are ignoring or the system ignores the change in tau H. On the other hand, if I record the chemosensory drive, and then I play that into the system and I speed it up or slow it down, then I can get the system to run slower or faster in a way that is controlled by the timing of the sensory input. So the timing of the sensory input seems to be, in the closed loop model, seems to be governing the timing of the breathing, as opposed to the timing of the intrinsic timing variable, which is the H gate. Okay, I'll skip through other parts of that slide. All right, now I apologize, this is a complicated looking slide, but it's worth going through it, okay, because it really shows what's going on. So there's two different manipulations we can do that I mentioned before. One is where we have a closed loop, closed loop system, but we fix the value of H. The other is when we have an open loop system where we fix different values of G tonic. And so we looked at all different combinations. So let's look in the blue here first. In the blue system, we fixed, so H is varying normally, as in the Butera-Winslow-Smith model, but we fixed the level of chemosensory input. Okay, so the drive is just constant. That corresponds to this situation here. Okay, so that's our open loop Butera-Winslow-Smith model. And as I showed you at the beginning, there's the quiescent region where there's no firing at all. There's the bursting region, which with normal looking bursts, and then there's the beating region where you're just firing a steady series of bursts of spikes with no gaps. So if I look at this blue, the patterns in blue here, what I'm doing is I'm changing the value of G tonic. The nominal value is 0.3, that's the usual value. And so if I look up here, what I see is I get this bursting pattern, so I get my limit cycle, and H is varying up and down during the limit cycle. If we look back here, right, H is going up and down like this. So I'm plotting H over the whole period. And so you see at 0.3, H is going up and down between these limits. So within this region where I have bursting, H is varying within this little wedge. If I make G tonic bigger, the variation of H gets smaller, but I have a limit cycle with bursting. Once I cross this boundary, the fluctuations in H are very, very small because I'm beating. And so the H just kind of fluctuates in a very small way. It looks like just a line. And in the quiescent region, of course, H is constant during the cycle. So we can flip things around now, and we can say, well, all right, what if we made, if we had the closed loop system, but what if we were in this situation here in red, where we made H constant, but we could make H constant at different levels, right? We could change H into a parameter. Instead of just being a slow variable, we could make it a parameter. Then G tonic is varying, and we maybe have bursting or maybe have another pattern. So in that case, there's again a region where we have bursting, and as we vary H. So for instance, if I say I fix H at 0.7 here, okay, so I make H 0.7, I get a bursting pattern where G tonic is going up and down between these two limits. So that would be like looking at this here and saying, okay, in this situation, how much is G tonic varying as I hold H fixed at a certain value and let the system run. So I get bursting and I get G tonic going up and down. And if I change the value of H, I get a different range or a different range, etc. So now if I look at the closed loop system, the norm without H being fixed, I'm going to have a loop where H and G tonic are both varying. And oh gosh, if we were in class, I would have everybody draw a sketch for themselves of where in this diagram they think the pattern of normal breathing would be happening. So during normal closed loop eupneic breathing, there's bursting. During the bursts, H goes up and down and G tonic goes up and down. And so the trajectories we could plot in the G tonic H plane somewhere. And there's an obvious hypothesis that you can make, which is if I fix G tonic, I have to fix it somewhere in this region to get bursting. So I would expect the combined pattern to be somewhere in here. And if I do it the other way and I fix H, then I have to be in this region to have bursting. So I would expect to be somewhere in this range, which means that I expect my bursting pattern to be somewhere there. Does that make sense? I'm not sure if it's complicated, but okay. So here's what we actually find. This black loop here is the closed loop eupneic breathing cycle plotted in terms of G tonic and H. And so it's not in this region. It's actually in this region where if you just fixed G tonic to a value in this region, you would be quiescent. Although if you fixed H in this region, you could still be bursting. So you go around like this. And so the closed loop normal behavior is happening in a parameter range where open loop, you wouldn't even be bursting at all. You would be totally quiescent. So all these lines of evidence tell the story that the mechanism of bursting in the closed loop system, which is analogous to the actual animal, is completely different than the mechanism of bursting in the isolated system for which Jeff Smith, got a very nice science paper in 1991 by demonstrating that you could get fictive breathing. But it's not working the same way. So it's really important, we think, to study the interaction or the dynamics of the central pattern generator in intact systems if you possibly can, because otherwise you're studying something which doesn't necessarily tell you how the intact system works. Okay, but I'll pause there in case there are questions. There's more to say, but I also see what time it is. So I think I've made my main point.
I have a short methodological questions. For all the different components of the model, did you run additional experiments, or it's all based on the known physiology of the breathing system so far?
So Chris Wilson was able to do an experiment in which we did a couple of things. So Chris and Casey set up a dynamic clamp experiment in which we simulated the Butera-Rinzel-Smith model. And I'm sorry, let me rephrase that. With a pre-Butzinger complex slice, we simulated the parts of the system that represent the lungs and gas exchange and the chemosensory feedback. So the other parts of the model that we added to make the closed loop, and injected the corresponding current representing G tonic into the cells in the pre-Butzinger complex. And we're able to show that we got the eupneic breathing, the normal breathing patterns through that closed loop model in a way that was consistent with what we see in the computational model. So that was a tricky experiment to set up. They got it to work. It was published in a a conference proceedings of the Biomedical Engineering Society. We did not get it to work, 30 times, and we didn't publish it in a full version of it, like in the Journal of Neuroscience. So it's been sort of, it's been confirmed in a limited way. The other, as part of those experiments, though, Chris Wilson dosed the cells with cyanide, and what cyanide does is it interferes with oxygen metabolism at the level of the mitochondria. And so he gave the cells the experience, so to speak, of hypoxia. And the way that they responded was by making a great big burst of activity and response, which is something that I haven't shown you yet, but is also a prediction of the behavior of the model. So we did not go experimentally through all these different regions and somehow test this picture experimentally. That wasn't possible. Does that answer the question?
Yes, thank you.
Okay. For the sake of time, I will skip this next bit. There, as one last piece of evidence that the underlying mechanisms are different, you can identify the kind of bifurcation that controls the onset and termination of the bursts. And there are different bifurcations in the case of the open loop versus closed loop. But that's also complicated to go through in detail, so I could come back to that if anybody wants. Okay, so another thing that we discovered in working with this model, and this is something that I think Frederick asked about earlier, is it has bistability. So you can, with the same parameters, depending on different initial conditions, you can either produce the Upnake pattern, but you can also produce the beating pattern. And what happens is, if you get into the beating pattern, it's very inefficient at driving gas exchange, right? The lung volume just fluctuates a very tiny amount. So you don't get fresh air into the lungs, you suck all the oxygen out, and so here you're down at 50 millimeters of mercury, instead of normally you're fluctuating around 100. That means that G-tonic, which increases as the blood oxygen level decreases, G-tonic goes up. And so you drive the system harder and harder. And as a consequence, you continue to have poor gas exchange. And so you get this negative feedback loop, and you can get stuck in this tachypnea-like state. I have a video that actually shows a patient with tachypnea. It's about 2 minutes long. I could show it if there's any interest, but I'm also conscious that we're getting to the end of the hour. So would anyone like to see a clinical example of, yes, so Peter Rowett. Okay, so to do that, I'm going to stop sharing that screen. The movie is not on my iPad, it's over here on my laptop, but that will just take a second to switch over. Okay, can you see my the slideshow again?
Yes.
All right. This is a training video. It might be 3 minutes long. This video demonstrates some of the signs of respiratory distress in an infant, including tachypnea. So this was a patient with pneumonia. And what you'll see here is the rapid shallow breathing. So 92 breaths per minute is quite large, even for an infant. So this is not really what's happening in our model. In our model, the spikes are happening, much faster than this. And this infant is getting enough gas exchange, that they're still alive. But, like tachycardia, which is excessively rapid heartbeat, tachypnea is excessively rapid breathing. Okay. So I will switch back so I can keep writing on my screen. Here we go. All right. I will, we can do fast, slow analysis on the model. I won't bore you with that. Maybe I'll show you this last piece of it. So in fact, I'm just going to focus on this. There you go. I can hide the parts. Oh, that's lovely. Hide the parts I don't want to talk about. So here we have a parameter in the model that represents the metabolic demand. It is the rate at which oxygen is pulled out of the blood and into the tissue for whatever purpose the tissue is using it. So we can vary M. If M is bigger, then we're asking more of the system. We can take the Butera-Rinzel-Smith system and drive it without any sensory feedback. That's the open loop model. And then it just produces exactly the same limit cycle, the lungs open and close, and we can increase the metabolic demand. And what you see is there's a nice steady decay in the blood oxygen level as you increase the metabolic demand. Because there's no sensory feedback, you just keep producing exactly the same respiratory pattern. This is like someone who's hooked up to a ventilator where the ventilator is set on autopilot. And if that person could somehow get up and start jogging, you know, they would run into trouble because the blood oxygen or the breathing system wouldn't respond in the way that yours or mine would. If you make the system closed loop, though, now you're sensitive to the chemosensory input. And now what happens is, as I increase the metabolic demand, this was the level about here that we had in everything I showed you before, or maybe it was 0.6, I forget, somewhere over here. As you change the metabolic demand, you don't make a very large change in the blood oxygen level. To me, this is analogous to homeostasis that you would see more famously in something like body temperature. If you look at a plot of, there are experiments in which they take small mammals and they put them in a temperature controlled chamber and they measure their internal temperature, and you know, warm-blooded creature keeps its body temperature more or less the same until it can't anymore. And then it dramatically goes up or it dramatically goes down. You're either hypothermic or hyperthermic. And physiologists have dubbed this the chair phenomenon. So you have this plateau region, and then on one side you go up and on the other side you go down. Here we have a similar kind of chair, which is to say there's a broad region over which the system makes adjustments in order to keep the quantity of interest, namely the average blood oxygen, keep it as constant as you can, but you pay a price. Eventually there is a catastrophic collapse. And that is, in our model, that's because you get past this border into tachypnea. Okay. I can just tell you, I don't have a figure for this because it's so new, but if you recall, oops. The sigmoid with G-tonic and concentration of oxygen concentration in the blood. The sigmoid is described by several parameters. Now there's a hypothesis floating around the clinical COVID community that the mechanism responsible for these silent hypoxemia patients is a reduction in gain in the chemosensory feedback pathway. I think there are other hypotheses, but this is one that we can address in our model. So what would a reduction in gain mean? Well, there's a couple things that could happen. You could just have a shallower slope for the sigmoid, or you could shift the sigmoid to the right or to the left, or you could change the height, right? It's got the sigmoidal transfer function has got 3 parameters in it, controlling those 3 aspects. So we've been playing around with this and asking, well, what happens if we change one of those in the model. And so far, we've been able to find changes that will either accelerate or delay the collapse. Or we found a parameter change that raises the steady state level, but then the collapse comes much sooner. And we found a parameter change that lowers it not down to like 50, which is what they're seeing in the clinic, but lowers it down significantly. But then the collapse is delayed. So this would be as if someone could have lower blood oxygen, but sustain a much higher level of exercise. None of this makes sense yet. So we're still looking at what combination of parameter changes, if there is one in this model, would give us something like, you know, a reduced sustained lower hypoxia or lower oxygen level. There are reports that in these same patients, the CO2 exchange is basically unimpaired. And it's the CO2 level increasing when you're not breathing well that makes you feel like you're not getting enough air. That's when you're the sensation of, it's called dyspnea or air hunger. That's largely due not to directly being aware of hypoxia, but being aware of elevated levels of CO2 when you feel out of breath, for instance. That's not happening. So that path, and the CO2 is somehow getting out through the alveoli, but the COVID virus is attacking the cells that produce the surfactant that inflate the alveoli, and somehow that's impairing oxygen exchange more than CO2 exchange. And so they're becoming hypoxic without experiencing the qualia, the feeling of being short of breath. So anyway, that's something that we're hoping to push this model to a better understanding of in the very near future. All right, well, that's the hour. There's more stuff to say about, oh, there's one last thing. Okay, so yes, there are different mechanisms. I kind of said that. I'm excited because we just got a paper accepted, like just got it accepted, that in which we develop mathematical tools for studying how you can compare the limit cycles when you change parameters. The problem is if you change a parameter like metabolic demand or something, you change both the period and the shape of the limit cycle. And as you know, looking at it from a control point of view, it's tricky to take apart how those things react with each other. So we have a way of doing this now, which we think is quite novel and exciting. That is, I mentioned last time, shape response curves. And the paper's on the archive, but it's going to appear soon in press in SIAM Applied Dynamical Systems. And if anybody's interested in that, I would be happy to stick around longer and say a little bit about that. But I realize some people may need to go, so I will stop with my conclusions here. And thank you for all for your attention.
Peter, could I ask a question?
Please, Chuck, go ahead.
We know that the mind and the practice can change the pattern of breathing. How does it play in the model?
Are you talking about meditation or
yeah, either meditation or just even just a deep breathing practice? We can
deep breathing practice. So the model is deliberately very simple, only 7 dynamical variables. And the only parameter that really has to do with, let's say, the interaction of the cardiorespiratory system with the rest of the body is this parameter M, which is the metabolic demand. So if through relaxation, meditation, or other behavioral activities, you reduce the metabolic demand, your body gets by with less oxygen, that would correspond to pushing you, shifting you up in this direction in the curve. But I think that's probably not a richly parameterized enough model to really get at the interesting aspects of things like deep breathing. This model doesn't do anything about sighing or augmented breathing yet either, which is something that I mentioned at the beginning that we're interested in understanding. There might be a connection with deep breathing. Some of the more sophisticated models that were published about the same time that are based more on CO2 sensing use more than one respiratory area. So instead of just the pre-Bustringer complex, there are a couple of other nuclei that are interacting with it, each of which has its own kind of built-in oscillator. And so the pre-Bustringer complex generates the inspiratory rhythm, but there's also active expiration, and there's a region that's active during sort of at the end of inspiration and during expiration. And so people have built models. John Rubin, for instance, has got models with so-called three-phase rhythms, where it's like you breathe in, you breathe out, then you kind of wait, then you breathe in again. And so there's these three distinct phases, which is actually pretty similar to in the Aplysia model, if you were here last time, where there's protraction open, protraction closed, and retraction, three different pools trading off 1, 2, 3, 1, 2, 3, 1, 2, 3, and then modulated by sensory feedback. So deep breathing, you could also, I guess, look at the strengthening the diaphragm muscle in the model, like the same neural activity could lead to a larger lung expansion. So with the physical practice and exercise of breathing, that might happen. Certainly one thing that happens with patients on ventilators is if they're being mechanically ventilated for an extended period of time, like a week or more, and the diaphragm muscle is not being activated, you lose muscle mass rapidly. And this is a mechanism that can lead to ventilator dependence, where just by not actually breathing actively for a week, you lose the ability to breathe, even if the central control pathway was intact. And that's pretty bad. So, okay, there's questions in the chat too. Leaf asks, the model seems vulnerable to getting stuck in a beating regime. Did you think real animals have mechanisms to protect against this? Yes. So I wouldn't take this model too literally for a couple of reasons. One is that there are mechanisms involved in regulating the breath by breath action that are not based on chemosensation, but are based on things like mechanical stretch receptors, pulmonary stretch receptors. You breathe in, and there's a reflex called the Herring-Brewer reflex, which one of the things it does is it protects you from breathing in so much that you damage your lungs. So when the lungs expand by a certain amount or at a certain rate, there is a reflex that shuts off the inspiratory drive. And so one possibility, and actually this is what we're hoping to explore in the project we proposed recently, is that there are multiple layers of control, the lowest layer being sort of neuromechanical, where things like, without having to involve chemosensation, there's mechanical control at the breath by breath level. The chemosensation may come in at another level that's sort of wrapped around that lower level, where if there isn't any chemosensation in the model, then you can you breathe as if it were open loop, but you will gradually drift out of the right range for normal oxygen levels. And then the chemosensory pathway is going to kick in. And maybe that's what is causing these augmented breaths, that you sort of start drifting into hypoxia, and then you take an extra big breath that resets the system and gets you back where you need to go. So that sort of thing may be happening. We also don't have CO2. So if I was stuck in the hyperdrive region, my CO2 would be building up, and there can be interbraided pathways. Since breathing is so important, you're going to expect to find multiple redundant pathways that work together to make it more robust. So I don't think that we see things like tachypnea in animals, but I mean, I don't know the veterinary literature. So yes, I do think animals have mechanisms to protect against this. And second question from Leaf, how quantitatively well characterized is the pathway from chemosensation to conductances? It is not as quantitatively characterized as we would like. So the sigmoidal feedback is a guess on our part. There are not, that's a hard experiment to do, because what you have to do is control the blood gases while measuring the input, both in this system and in the Aplysia system, and in, I think, in many neuromechanical systems. The hardest thing to measure is the sensory feedback, right? Because what is the experiment? You have to manipulate the condition of the body, of the plant, of the external aspect, and then you have to measure the signals coming back. The motor signals are really big because they're driving muscles, but the sensory feedback signals are, they tend to be carried by a large number of very small axons, which makes it hard to measure at the, you know, on route, or hard to measure at the target. I mean, we're hoping to come up with ways to do that, but at the present, that's where there's a big, that's one place where there's a significant gap. Okay, thanks, Leaf.
All right, thank you very much for this amazing two-part talks. I think if no one has any more questions, we're going to close now.
Yes, I appreciate the chance to talk to everybody for so long about this, and thank you for your attention.
We always appreciate closed-loop systems.
Yeah, I think they're very important.
No, that is very nice combinations of experimental techniques, modeling, and thinking. I don't know. It's really really nice. Thank you again.
Okay, thank you.
Thank you, everyone. See you next week. Bye bye.